Hello my dear students. Now in this video we are going to discuss TCA cycle, which is also known as Krebs cycle and which is also known as you can say citric acid cycle. So this has three synonyms. One is TCA, tricarboxylic acid, second is Krebs cycle, and third is it is also known as citric acid cycle. Now again we are going to follow the same pattern as we are following in the rest of the pathways or cycles. First of all, let's write it TCA. It stands for tricarboxylic acid, also known as Krebs cycle because it was discovered by Sir Hans Krebs. Discovered by Sir Hans Krebs. And it is also known as citric acid cycle because the first stable product is citric acid. So now in this video we are going to discuss this TCA, and always remember this is a very important cycle. The reason being that the most important point about this is, जैसे we've been studying, we studied glycolysis. I told you it was a catabolic pathway. Okay, TCA is amphibolic in nature. It is amphibolic. Very important. Means it can act as a catabolic one also and as an anabolic one also. Second important point about this is, again we will come to those same things which we are discussing in other pathways. What is the site for TCA cycle? Now the site is, it occurs in the mitochondrial matrix. So it occurs in the matrix of mitochondria, or you can say mitochondrial matrix. Now one very important aspect of this is that all the enzymes of citric acid cycle are present in the matrix of mitochondria except succinate dehydrogenase. So all enzymes are present in the matrix of mitochondria except SDH, and this SDH stands for succinate dehydrogenase, which is present on the inner mitochondrial membrane. This is very important to remember, which is present on the inner mitochondrial membrane. Right, so this is second point which we have to remember. Then if we talk about the energetics, so energetics is that it gives one molecule of acetyl coenzyme A gives 10 ATP molecules. 10 ATP. And then we know that we started with one molecule of glucose forming two molecules of pyruvate, so automatically two molecules of acetyl coenzyme A will be there. So two molecules of acetyl coenzyme A will give 20 ATP molecules. This we are going to again study in detail when we will study the cycle. This is energetics. And if we talk about the regulation, there are three enzymes, very important enzymes, which play a very important role in regulating the entire TCA cycle. One is citrate synthase, obviously which is the first step, and it has to be irreversible. Okay, so one is citrate synthase. Second enzyme is isocitrate dehydrogenase, and the third enzyme is, which is again an example of multi-enzyme complex, alpha-ketoglutarate dehydrogenase. So these are the three regulatory enzymes of TCA cycle. Okay, so we have studied the site, we are going to study the energetics, and then we are going to study the regulation. So since it is a cycle, so what is the first substrate of this cycle? So I would say the first substrate is oxaloacetic acid. First substrate of TCA is oxaloacetate. And now what happens is that this oxaloacetic acid, which has 4 carbon atoms, this will combine with acetyl coenzyme A, which is obviously we know what is the source of acetyl coenzyme A, which has 2 carbon atoms. It will combine to form citric acid or citrate, which has 6 carbon atoms. Right, 4 plus 2, 6. So this is simple condensation reaction. 4 carbon atoms combined with 2 carbon atoms forming citrate. And the name of the enzyme here is citrate synthase. Now, this citric acid will give out a molecule of water and it will form cis-aconitate, and it will take a molecule of water forming isocitrate. So you can say water molecule given, water molecule comes. Now what happens is this isocitrate will undergo first decarboxylation reaction. Mane, it will give out a molecule of carbon dioxide. So isocitrate will, iso means it is an isomer of citrate only, and it has again 6 carbon atoms. When it gives out one molecule of carbon dioxide, it will be converted into alpha-ketoglutarate, which has 5 carbon atoms. We have given one molecule of carbon dioxide here. Okay. So if we talk about the enzymes here, I can simply say that here the enzyme is ICD, which stands for isocitrate dehydrogenase. I'll write the full forms later. Here I am just writing it as ICD. So dehydrogenation hai, so by default NAD will be converted into NADH plus H positive. Now next reaction is this alpha-ketoglutarate will give one more molecule of carbon dioxide, same oxidative decarboxylation, and it will be left with only 4 carbon atoms, and it forms succinyl coenzyme A, where the enzyme is alpha-ketoglutarate dehydrogenase. So whenever dehydrogenase is there, automatically we have to write NADH plus H positive. Now the number of carbon atoms have been given out, and that is what is the target. One is here, one here. And acetyl coenzyme had only 2 carbon atoms. That means complete structure, complete you can say 6 carbon atoms of glucose are dismantled. Now we have to convert this succinyl coenzyme A into oxaloacetic acid. So this succinyl coenzyme A then first of all will form succinate, which will form fumarate, which will form malate, and this malate will form oxaloacetate. So this is how the entire TCA cycle will be there. Just okay, malate. Now the question is, what are the enzymes involved in this? This succinyl coenzyme A, when it is being converted into succinate, it is forming GTP here. So GTP will be produced from GDP, which is coupled with ATP. And the name of the enzyme is succinate thiokinase. Automatically succinate to fumarate, the enzyme is succinate dehydrogenase. Dehydrogenation, FADH₂ will be given out. And this is the only enzyme which is present on the inner mitochondrial membrane. Just keep this thing in mind. FAD₂, FADH₂, the enzyme succinate dehydrogenase, which is present on the inner mitochondrial membrane. Then fumarate, 2-malate, they all are linked as actually the isomers. So I can simply say that fumarase. And oxaloacetic acid is produced by dehydrogenation reaction. So I can say that again the enzyme is malate dehydrogenase, and NAD will be converted into NADH plus H positive. Now looking at the enzymes, I'll just highlight the enzymes. One is isocitrate dehydrogenase, succinate thiokinase, alpha-ketoglutarate, succinate dehydrogenase, fumarase, malate dehydrogenase. And the most important enzyme, citrate synthase. Here we can simply say that here cis-aconitase is there. Now coming over to this, I'll just write first of all, first enzyme ICD, which stands for isocitrate dehydrogenase. Second enzyme is alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase. Then comes ST, which stands for succinate thiokinase. Succinate thiokinase. Then comes SDH. SDH I have told you earlier also, it is succinate dehydrogenase. And always remember, succinate dehydrogenase is the only enzyme present on the inner mitochondrial membrane of the mitochondria. Very important. Only enzyme present on the inner mitochondrial membrane. Then if we talk about the other enzyme, fumarase, fumarase is clear. Then we come over to MDH, which is malate dehydrogenase. So these are the important enzymes of the entire TCA cycle. Clear? Now after this, then we come over to again the energetics. Now if we talk about the energetics completely, I can simply write it also that there are 3 FAD. If you see here in the cycle, this is 1 NADH plus H positive, this is second, and this is third. So I can say that it is giving 3 NADH plus H positive, 1 FADH₂, and 1 GTP. So this becomes the energetics from one molecule of acetyl coenzyme A. So if I will just write it directly, as you can say that 3 NADH plus H positive. So it will be equivalent to 3 into 2.5. 1 molecule of FADH₂. So it will be 1 into 1.5. And 1 GTP, which is in terms of energy equivalence related to ATP, so 1 into 1.0. So if we count this, this comes out to be 7.5, 1.5 and 1.0. 10.0 ATP, and that is what it is all about. So this is from one molecule of acetyl coenzyme A. So if one molecule of acetyl coenzyme A is giving 10 ATP, I can simply say that 2 molecules of acetyl coenzyme A will give 10 into 2 equivalent to 20 ATP. Okay. And if you can say complete the entire energetics, I can simply say that we started from, because TCA cycle is again the dead end. Because the complete 2 carbon atoms which were present in acetyl coenzyme A, they are completely dismantled. So I can say that one molecule of glucose having 6 carbon atoms is going to form 2 molecules of pyruvate having 3 carbon atoms. 2 molecules of pyruvate are going to form 2 molecules of acetyl coenzyme A having 2 carbon atoms, and this acetyl coenzyme A will undergo this TCA cycle like this, giving out 1 carbon dioxide here and 1 carbon dioxide here. So these 2 molecules of acetyl coenzyme A will give 1 carbon dioxide, 1 carbon here, 1 carbon here, and the entire structure will be dismantled. So I can say that, and this is how the entire oxidation of this glucose is taking place. Now if we calculate the entire energetics, I can simply say that 7 ATP are produced here, here 5 ATP, and here if we talk about total, there are 20 ATP molecules, and overall 20 plus 7 plus 5 is equivalent to 32 ATP molecules. So I can simply write it like this, that 1 molecule of glucose on complete oxidation gives 32 ATP molecules. This is very important, which we have to learn. Right? Now, this is complete energetics. Then we come over to the most important part, I would say, again after energetics, the substrate level phosphorylation, because the same process I mentioned in glycolysis where there were 2 steps of substrate level phosphorylation. So if we talk about TCA cycle, first of all, what is this substrate level phosphorylation? Now here also, if we have to define, I can simply say that substrate level. What is phosphorylation? It is simply attachment of inorganic phosphate. From where that phosphate is coming? It is coming at the expense of the substrate. So how do we define? Simply you can say that addition of phosphate at the expense of the substrate. Right. And here there is only one reaction which is actually an example of substrate level phosphorylation. And if we talk about the reaction is succinyl coenzyme A is converted into succinate. And here coenzyme A, which is SH-CoA, so we always write it is a thio group. So the enzyme here is succinate thiokinase. And what happens here is simply GTP is synthesized. And the importance of this reaction is that when GTP is synthesized, it is coupled with ATP like this. So in terms of energy equivalence, GTP and ATP are the same, so we just count it as 1 ATP only. But since it is coming from the substrate, this is substrate level phosphorylation. Then second important point is regulation. Now if we talk about the regulation, we know that citrate synthase, the first enzyme, It is catalyzing irreversible reaction. And if we look at this reaction, we can simply say that acetyl coenzyme A is coming like this, and this oxaloacetic acid is forming citrate. And here the enzyme will be citrate synthase, which is governing this acetyl coenzyme A. Right. And the second enzyme, if we say, in fact, the rate limiting enzyme is isocitrate dehydrogenase. Isocitrate dehydrogenase is the rate limiting enzyme of this reaction, which is simply converting ICD, or this is the enzyme, I'll just write, it will convert isocitrate in the presence of ICD to alpha-ketoglutarate. Here there are 6 carbon atoms and here there are 5 carbon atoms. So this is actually oxidative decarboxylation also. Not exactly, I would say कि it requires TPP. यहाँ पे the most important thing you have to remember is, एक तो चलो this is there, along with this it has dehydrogenation also. Very important. Okay. And the third important enzyme, which is catalyzing a physiologically unidirectional step, it is alpha-ketoglutarate dehydrogenase. Very important. Full form is simple, alpha-ketoglutarate dehydrogenase, and it is catalyzing physiologically unidirectional step. Unidirectional step. Basically, where what happens is another decarboxylation reaction occurs. So this is the reaction. Alpha-ketoglutarate will be converted into succinyl coenzyme A, giving out again carbon dioxide, and NAD is converted into NADH plus H positive. Now the most important point which we have to remember here is that alpha-ketoglutarate dehydrogenase, it is an example of multi-enzyme complex. It is an example of multi-enzyme complex. Now, what is the difference between PDH and this? Now, in my previous video, I had clearly mentioned that PDH always catalyzes a reaction in which simple pyruvic acid is converted into acetyl coenzyme A, and it is converted into active and inactive forms. So, if there is an increase in ATP to ADP ratio or increase in NADH to NAD ratio, PDH is always inhibited. Here, the thing is that structure is the same, but alpha-ketoglutarate dehydrogenase cannot afford to undergo regulation. Very important point, right? So, I can simply say that first of all, it is an example of multi-enzyme complex, and the most important, I would write it also here, structure similar to PDH, same, like B1 is there, B2 is there, B3 is there, B5 is there, and lipoic acid is there. And along with this, it has 3 enzymes, E1, E2 and E3. But the only difference, and B1 is the main one, but the only difference is it cannot be converted from active to inactive or vice versa. Active to inactive or vice versa. because the body cannot afford to undergo regulation of this TCA cycle. Inactive nahi hoga. Why? Because TCA cycle is that cycle which is operative all the time. And this is the major pathway of oxidation of glucose. And there is again, there is an intermediary metabolism occurring in our body, whether we are having fats in the diet, whether we are having proteins in the diet. They have to be converted into TCA cycle one way or the other. So the ultimate pathway of degradation of all the respiratory foodstuffs is TCA, which is linked to electron transport chain, which is linked to synthesis of ATP. And overall, we call this terminology as biological oxidation. So the body actually cannot imagine that TCA cycle will stop for some time. Still they are regulatory, but it will not be stopped. And that is why we always say that alpha-ketoglutarate dehydrogenase will always catalyze the reaction, oxidative decarboxylation taking TPP, but it won't be converted into inactive form. So this is what is the regulation. Then another important point which I told you about this is that it is an amphibolic pathway. So TCA is an amphibolic pathway. Now what do we mean by this amphibolic? That simply you can say that it has two roles to perform. One is anabolic, the other is catabolic. Okay, now what is the difference between this? Catabolic, I can simply say, as you saw also, it is forming carbon dioxide, and later on you will see it is leading to the formation of water also. Now coming over to the anabolic role, the most important role played by TCA cycle is that it undergoes lipogenesis. It is required for the synthesis of fats, one thing, and it is linked to the amino acids also. Right. And if we talk about lipogenesis, basically what happens is, I'll just give you a hint here that lipogenesis always occurs in the cytoplasm. Right. And precursor is lipogenesis or fats ka precursor is acetyl coenzyme A. And this acetyl coenzyme A is present in the mitochondria. So acetyl coenzyme A is impermeable to mitochondrial membrane. Instead, citric acid is transported from the mitochondria to the cytosol, where it is converted back to acetyl coenzyme A. And this is also known as citrate shuttle. So if TCA cycle is not there, citrate shuttle is not possible. So basically what is happening is, say this is the mitochondria and this is the cytoplasm. So what is happening is that this acetyl coenzyme A is combining with oxaloacetic acid to form citrate. Right? Now this acetyl coenzyme A is impermeable. So this citrate will travel like this, and this it will be converted into the products from where it is produced. So it is going to form acetyl coenzyme A here and here oxaloacetate. And here the name of the enzyme is ATP citrate lyase. And in fact this acetyl coenzyme A will now be utilized for the synthesis of fats. Acetyl coenzyme A is a precursor of cholesterol also. So this acetyl coenzyme A will be transported via this citrate shuttle. It's like a game of badminton. Citrate shuttle. So this is a very important anabolic nature of this. Then if we talk about oxaloacetic acid, this oxaloacetic acid can easily be converted into aspartate. right? So this is one very important feature of citric acid. Then after this, then we come over to the inhibitors of TCA. So if we talk about the inhibitors, the most important inhibitor here is fluoroacetate. Fluoroacetate. So what will happen is this fluoroacetate, basically the reaction is what? Oxaloacetic acid is forming citrate. This is the reaction we have studied. But when fluoroacetate comes, it is going to form fluorocitrate, thereby blocking TCA. So this is the inhibitor of TCA cycle. And the most important feature of this TCA cycle is it plays a very important role in intermediary metabolism. Intermediary metabolism. So what is happening here is, now we say that this is acetyl coenzyme A. This acetyl coenzyme A is combining with oxaloacetate to form citrate. And here we can say that the chief intermediate is alpha-ketoglutarate and here succinate is there. Now the most important thing here is that this acetyl coenzyme A is required for the synthesis of fats. It is required for the synthesis of cholesterol. It is required for the synthesis of ketone bodies. Right. So this is occurring in the fed state, and during starvation also fats are broken down to form acetyl coenzyme A. Similarly, oxaloacetic acid is converted into aspartate, which itself is an amino acid. Similarly, alpha-ketoglutarate is produced from majority of the amino acids. In fact, except 3 amino acids, 17 amino acids undergo transamination forming alpha-ketoglutarate. So it is like all the amino acids through transamination require this. And if there is decreased concentration of alpha-ketogluteric acid as it occurs in hyperammonemia, what happens here is that TCA cycle is the one which is depleted. Then this succinate, or you can say succinyl coenzyme A, if we talk about this succinyl coenzyme A, it is being produced from propanol coenzyme A. And what is this propanol coenzyme A? It is coming from the oxidation of odd chain fatty acids. So you can see that TCA cycle, how important this is. Then coming over to this, I told you it has 3 NADH plus H positive reactions. It has 1 FADH₂ reaction and 1 GTP. So what is the source of this proton, or what happens to this hydrogen? And since it is occurring in the mitochondria or the matrix of mitochondria, these protons are actually carried by electron transport chain in the presence of electrons, in the presence of oxygen, to form this water. And this water so produced is known as the metabolic water. And whatever energy is being liberated at every step, this energy is utilized to form ATP. So this electron transport chain and the synthesis of ATP are two coupled processes. Synthesis of ATP is known as oxidative phosphorylation, and this combined is known as biological oxidation. So this is the importance of TCA cycle. Okay. So overall, if we have to summarize TCA cycle, I can simply say that it is the major pathway of oxidation of glucose. One molecule of glucose which enters via glycolysis, via link reaction, enters TCA, thereby totally producing total how many ATP molecules? 32 ATP molecules. Okay. Where it is occurring? It is occurring in the matrix of mitochondria. All the enzymes of TCA cycle are present in the matrix of mitochondria except succinate dehydrogenase. Because succinate dehydrogenase is having a very low redox potential. So it is present in the inner mitochondrial membrane. Then second point is that there is only one reaction of substrate level phosphorylation in this, where succinyl coenzyme A is converted into succinate. Here GTP is produced, whose energy equivalence is basically equal to that of ATP. Third important point about this is alpha-ketoglutarate dehydrogenase, which is catalyzing the oxidative decarboxylation, has the structure similar to PDH. The only difference is that it is not present in active and inactive forms. And since it is catalyzing the oxidative decarboxylation, the coenzyme required for this is vitamin B1. So this is all about TCA cycle. And here we finish up with the major pathway of oxidation of glucose, glycolysis to link reaction to TCA. And this TCA, you will be seeing that whatever metabolism we are studying, whether it is proteins, amino acids or fats ka metabolism, it is ultimately, you can say, integrating into TCA. And that is why there is a very important terminology which exists in biochemistry, intermediary metabolism. And who's the hero of this intermediary metabolism? It is none other than our TCA cycle. And this is the only amphibolic pathway also. And I gave you the reasons that why it is known as an amphibolic pathway. Okay. So I hope you have understood the TCA cycle in detail. Thank you so much.